Good evening and welcome to our special 2020 Shaw Prize Awards. The Astronomy Award this year goes to Professor Roger Blandford. His work has led to a greater understanding of active galactic nuclei. He's also expanded our understanding of black holes and massive energy bursts called relativistic jets that are produced by spinning disks of gas and even stars surrounding these mysterious objects.
Stanford University, an active scholastic and experimental nuclei of astrophysics, where observations of larger cousins, called active galactic nuclei, have provided greater insight into the origins of the universe. Work by this man, Roger Blandford, has shed more light on the role of black holes in these galactic furnaces. Blandford arrived at Stanford in 2003 to begin working at the Kavli Institute for Particle Astrophysics and Cosmology as director. Kavli continues a decades-long advance using steadily improving new technology applied to the ancient science of astronomy.
I have to say that I did not expect it would last. I thought there would be a sort of flurry of excitement and people would make discoveries and find out things, and then it would just all get too hard, and then, essentially the field would plateau, and boy was I wrong. That process of the application of technology followed by discovery, followed by interpretation, has kept on going, and it's still happening today.
Active galactic nuclei form in the centers of galaxies. They include a black hole as well as gas and stars. Active black holes form quasars and together create a lot for astronomers to look at.
Most of them do seem to have a black hole. In some cases, these black holes are what we call active, which means to say there's a lot of light and radio waves and X-rays and gamma rays that are observed from it or from around it. In others, they seem to be dormant. And in the case of the center of our galaxy, there is a 4 million solar mass black hole there, and it is very dormant, it is very inactive, and for that we should be rather grateful, because we wouldn't want to live right next door to a quasar. The environmental impact would contravene various federal regulations.
Again, very good questions.
A teacher of astrophysics for decades, Blandford never shies away from using humor to make a point about the science of black holes.
This is not the European debt crisis. Thank you very much.
One of the wonderful things about Roger is he's knowledgeable about almost everything, but he comes to everything with a really generous spirit, I think. And he's always curious. He's always really interested in learning new things about almost any topic. Founding a new institute in astrophysics at Stanford is one of those really foundational contributions. He really got the institute off the ground. We're now 17 years old and quite robust as an institution, but it was really Roger's vision to make that happen. And one of the key points of that vision was to sort of develop an institute which actually brought together the kind of theoretical ideas that Roger and others have worked on in particle astrophysics, in understanding how high energy particles work, how black holes work, and also in cosmology, and to bring that together with an institute that it could actually build things.
Roger's done, foundational work in cosmology with gravitational lensing. He's done remarkable work that I first learned about black holes, and not only how they swallow matter, but from the point of view of gamma ray astronomy and astrophysics, how they interact with matter and are able to accelerate it to extraordinary energies and actually extract energy out of a black hole. And that's a tremendous insight that goes back many years. And we're still learning about how that all works.
Look at this black hole. One of the things that happens in a quasar is that we have a disk of gas that is orbiting around it. So if we imagine having a disk of gas spinning like that, orbiting around a black hole, so it'll orbit around the black hole like that, then the gas that's in this disk will have a bit of friction in it, and that friction will cause it to spiral inwards, just a little bit like a satellite in the Earth's atmosphere. And as it spirals inwards under this friction, just like having friction in your when you rub your hands together, things are going to get rather hot. And this gas will get very hot, and it'll radiate X-rays and ultraviolet radiation and so on for the gratification of observational astronomers. So we can see these quasars from the radiation that these accretion disks produce.
Blandford says as black holes spin, magnetic field lines will take rotational energy out of the black hole. Energy responsible for these large relativistic jets of energy observed flying out of these distant objects. Jets so powerful they nearly reach the speed of light.
If we want to know a little bit more about how that must happen, the one thing you need to know about magnetic field is that it's a little bit like a magnetic field line. It's a little bit like this bungee cord. It has a tension associated with it, and if we imagine it wrapping it round a jet like this, and take a second magnetic field line here and wrap it around the jet like this, then it can be like my belt, and it will hold me in place too. So it will provide a means of collimating and constricting the jet and making the structures that we observe with our radio and other telescopes.
Blandford hopes this will soon be confirmed in observations of an accretion disk around a massive black hole in the galaxy M87, 54 million light-years from Earth. It was the very first real picture of a black hole.
The normal view of this is what the radio telescopes are actually looking at is the inner part of an accretion disk that's swirling around the black hole close to it. And I'm exploring myself the possibility that it is in fact a magnetic field that is around the black hole that they're actually looking at. There is an accretion disk there, but it's at larger distances than what is being observed by the Event Horizon Telescope.
Roger takes a break from the planet-sized blasts of radioactive energy for a few sparks at the end of some birthday candles.
You can get to the flowers, Nagel.
Family is arriving for his grandson's birthday.
Properly socially distanced. Okay.
Liz, Roger's wife of 50 years, says they met when they were both on a summer study trip in England.
We met in the summer of 1970. We met at Hurstmonceux. Have you already told them this?
Nope,
No?
nope.
All right, we met at Hurstmonceux Castle. It was kind of a fairy tale meeting. Hurstmonceux Castle at that time was the headquarters for the Royal Greenwich Observatory. And it was a castle with a moat and swans. And it was one of those really rare English summers, sunny, dry, really magical.
It was, as Liz said, a very magical time, and it was a very happy time, and we've stayed in touch with one or two of our friends there. But I remember seeing Liz for the first time and being really struck by her, and that hasn't changed.
Marriage soon followed. They had 2 sons, both engineers now. Jonathan remembers his dad being humble about achievements.
I remember asking him when I was a kid what he did to get his position and get his doctorate, and he said, "Oh, I did a couple sums," is what he told us. He was never really forthcoming about how it was, and I found out later that he won awards for some of his graduate work, but he never really talked about that, just what he was doing next.
Edward thinks his sense of humor made him a better teacher of young astronomers.
In his line of work, being able to explain things and teach at all levels is very important, and a big part of that communication and the ability to convey complicated thoughts is to break things down and make it entertaining.
Roger was born in Lincolnshire and grew up in Birmingham, both in England.
It's about 4 miles away from where Isaac Newton was born, and I boast about this to my students and they are totally unimpressed.
More impressive would be when the astrophysicist of Stanford shed light on the biggest mystery yet to be solved in the universe.
I would say for me the #1 question is the identity of dark matter. As is well known, there's about 6 times as much dark matter as there is regular matter in the universe. We see it, as far as we know, just from its gravitational properties. It's a great puzzle in astrophysics, in cosmology, and in theoretical physics and in experimental physics. All of those are all looking at different aspects of the problem. And one hopes that over the next 10, 15, 20 years or so on, we'll know what the answer is, we'll know what it is, what its identity is.
Welcome back to the 2020 Shaw Prize Special. The Math Prize goes to David Kazhdan and Alexander Bailenson for their contributions to representation theory, the study of symmetries, both clear and hidden. Their work has been very useful in pure mathematics, as well as in the fields of quantum mechanics and network communications.
Jerusalem in Israel, where mathematicians at the Einstein Institute of the Hebrew University of Jerusalem dig into the deepest corners of pure thought. Mathematicians such as David Kazhdan, who began his career at age 14, publishing his first proof. He has amassed 60 years of scholarship, which all began in Moscow in the former Soviet Union as a stunt to skip school.
When I was 11, I decided that I don't want to go to school every day. I asked my parents to give me permission to skip 1 day of school. They say yes, if you do something reasonable. So I suggested them they'll learn chess. And for half a year I was learning playing chess. Then they said it is not sufficient, choose something else. So I've chosen mathematics. This is how I started mathematics.
He proved to be a protege.
Let me illustrate this by an early example when he was like a kid. I'm not sure exactly what age, but something like 18 or 19. He already became international famous for something which nowadays is called Kazhdan property T. What is amazing about this Kazhdan property T that it found so many applications in so many other areas, some of them by Kazhdan himself, but also by so many other people. And the most interesting is that they even found application, this property, into down-to-earth applied mathematics, and more precisely to computer science. There are nowadays some networks, communication, which are built by the so-called expander graphs. And expander graphs was, for the first time, when were constructed using Kazhdan property T's.
Colleagues describe Kazhdan as one of the last universalists in the field, where it's common that even people within mathematics don't understand each other's work. Hillel Furstenberg once had an office next to Kazhdan and remembers working with him on many problems.
I want to give a simple example of something called a lattice of one group, of one Lie group, and this is a lattice in another Lie group. And how do you know that they're not the same? Maybe they look different, but they might be the same. So using my notion of the group boundary, I worked pretty hard. One of the consequences of this theory was that these two groups are not isomorphic. They're not the same. They're different. Only afterwards I realized that if I use Kazhdan's, this notion of the Kazhdan property of a group, then it's immediate that these are not the same. I would have had a much simpler proof and a much shorter proof that these two are not the same.
Mistakes are made in mathematics, but what appealed to Kazhdan at school and later teaching at Harvard University was that once a notion has been proven, it will always be so.
It stays true, and its applicability to different facets of the world. I was always working in pure mathematics, did not ever work in applied mathematics, but it's pleasant to know that some of my works were useful for applications.
What's so special about him that he connects ideas and areas which seems to be completely unrelated to each other and make them into a unified type of theory and thinking.
He used to have, his door used to be next to mine. At that time, there was always a crowd in the hall. I would have liked to say, they all want to see me. No, they want to see David and consult him on some matter. But David is actually broad, not only mathematically. He's also broad in his interests. You can discuss history with him. You can discuss literature with him. In all of these, he's gone into these rather deeply. In fact, sometimes I make a joke. I'm not even sure he's a mathematician, because he's so intelligent, he would succeed in anything that he went to. He happens to be in mathematics.
Outside of mathematics, with the family, he tackled mountains and cycled roads with the same tenacity used to solve his theories. The children take his mathematics seriously, but not without a measure of sass.
Counting was never his strong point, but I'm sure he has other competitive advantages, and he certainly does. Having a mathematician as a father, I don't know if it's the mathematician, the scientist, the intellectual, but there's something to be said about having somebody who sets very high bar for himself, demands a lot of himself, and demands a lot from those around him.
7 years ago, David and his son were out attacking yet another road when a large truck hit David, causing life-threatening injuries. He was in intensive care for 2 months, but Kazhdan and his math endured.
I came to the Hebrew University the year he had the accident, and I hardly knew him then. And, you know, very short while after he was out of intensive care, people from the department would go and talk mathematics to him at the department, and I thought that was amazing. As if he just decided that his physical circumstances don't matter at all. I find that remarkable.
These days, instead of hiking and cycling, he and his son take long walks. He's a bit slower, but no less determined.
We as bike riders, we say, if you're not pedaling, you're on a downhill. And I think part of his way of living, always was, always is you got to keep on pedaling.
Our second math prize winner works here at the University of Chicago. Alexander Beilinson joined the mathematics department in 1998 after working for MIT. Like Kazhdan, Beilinson moved to the US from the former Soviet Union, which at the time had a successful record of grooming young mathematical talent.
I was to mathematical school, and it was really excellent. Some professors from the university taught classes there. And then I had near our summer house, well, a neighbor was actually also himself an excellent mathematician. And when I got interested reading mathematical books myself, I started talking to him. In mathematics, it's extremely important, because when you start to read books by yourself, your efficiency is extremely low.
He says he became a mathematician by chance. He says most young students who try to study eventually give up after a while, but he managed to persevere, and his colleagues say the field has benefited from his multiple contributions.
The area that I know him is the theory of motives. You may have heard of the Riemann hypothesis about the Riemann zeta function. That's one of the classic mathematical puzzles. And this is a vast expansion of that program. And it's an intermingling of algebraic geometry, so geometry and number theory. And that's the work of Sasha's that I know the best. And it culminated in the Beilinson conjectures that we all wrestle with now.
His working style can be as sedate as the family's pet snake, but then...
I don't know if you know somebody who has a fancy car, an extremely powerful car, but this car can also be driven at normal speed. And you are driving with your friend at normal speed, and then you ask a mathematical question. And suddenly you hear a rumbling from the engine, and you suddenly are going much, much faster than you were going a moment ago. So I would say that Sasha has two speeds, a very quiet, normal speed, and then a high speed, a math speed.
The family shares their father's love of nature and ecology. Today they're assessing a recent casualty with friend Jesse Ball.
Skull is crushed?
Yeah.
Oops.
What was it?
I think possum, no?
Oh, yeah, it's absolutely possum.
He didn't really push math onto us. We were just taught to be kind to humans and to animals alike and to plants, and that nothing was better than anything else, nothing was below anything else. We were taught to be curious, to ask questions, and to be curious and to be interested in what we're curious and interested about, and not what we're told to be curious and interested in.
We'll talk about things like animals and nature. We both share an interest in Zen, for instance, Zen Buddhism, which is to say just a curiosity in the hilarious predicament of being alive. So we both find that stupid and funny. And so there's a lot of laughing about that.
What does Beilinson think of the future of mathematics?
In truth, nothing changed. Well, people think in the same way as they thought before. Certainly they find some new thing, they do different research, but the essence did not change at all.
3 European scientists have won the Life Science and Medicine Award for the development of optogenetics, a revolutionary technique that's allowing neuroscientists to trace and control brain activity with light. Thousands of labs around the world are using the technique to unravel the complexities of the human brain and even develop treatments for brain diseases and mental health problems.
Controlling minds with light. What sounds like science fiction has been brought to reality by neuroscientist Gero Miesenböck. The Austrian scientist arrived at the University of Oxford in 2007. Here, his team can put flies to sleep and make headless flies take flight using nothing but light. Application of the optogenetics technique to manipulate an organism's neural circuits shines a light on how neurons drive behavior and intelligence.
I think that discovery is not just that you find an objective fact that's somewhere out there in the universe and you just record it like a passive observer. There's an element of creation also in discovering how nature works. And you conceptualize how you think a certain process actually runs. And that creative element is, I think, common to, for example, a writer, a novelist penning down a novel. I think it's very, very important not to suppress this artistic element in research.
Despite pioneering a whole new field of neuroscience, Miesenböck's childhood dream was not to be a scientist at all.
My dad was a classicist, and at some point I thought I might follow him into literature. But he advised me very strongly not to do that. Because, he said, the potential for discovery in the sciences is so much greater. You have an essentially unbounded universe of things that you can figure out. So I took his advice and became a scientist, but I've retained an interest in literature and also in writing. I think some critics might say that I'm still writing fiction when I write my scientific papers, but that's of course not true.
The 55-year-old gave up literature for medicine in university. However, born with the desire to create, he soon realized the repetitive work of a typical doctor was not for him. When he was finishing his medical training, he came across an unusual research paper, or what he called artistically presented scientific literature. He wrote to the author, James Rothman, and asked to join his lab at the Memorial Sloan Kettering Cancer Center in New York.
So my postdoc advisor, a fabulous scientist named James Rothman, who won the Nobel Prize in 2013, he was probably the dominant influence in my scientific upbringing, because he showed me that there is something like a personal style in science. One of the things that attracted me to the particular lab that I joined was that the scientific research was framed and presented in this artistic, aesthetically appealing fashion. That really attracted me. It was not just, as is sometimes dismissively said, stamp collecting, where people just observe and collect various observations. It was all driven by ideas rather than facts.
During his years with Rothman, Miesenböck worked on molecular cell biology, the science of how individual cells function. But he soon realized it would be far more interesting to study how intelligence emerges from the physical interactions of nerve cells. He began to develop genetically encoded tools such as light-emitting proteins to make neuron activity observable. One day in 1999, when he had already started his own lab, the idea to use light not only to observe but also to control the mind crossed his. He recalls the eureka moment.
The idea to optogenetic control came to me one Saturday afternoon while I was not really thinking about the problem, but I must have been sort of incubating it, and it suddenly sort of popped to the conscious surface like a bubble that reached the surface of a pond of water. I realized then and there that this could have a profound impact if it was feasible. So the next difficulty then, of course, was to try and find a genetically encoded receiver that could take a light signal and transduce it into electrical activity. And then it also became logically clear where one could find such genetically encoded receivers, and that is, of course, among the light receptors in our eyes and in the eyes of other animals. So the idea then was to take one of these light receptors and transplant it genetically from a light-sensing cell in the eye to a different cell deep in the brain. And if illumination of that cell deep in the brain then caused electrical activity, just like illumination does in the eye, then the approach would succeed.
The technique, when applied to specific neural circuits in the brain, would enable scientists to find out in unprecedented detail how each cell type contributes to our thoughts, emotions, and behavior. It took Miesenböck about 2 years to bring his idea to reality. This video captured the first use of optogenetics to control animal behavior. Cells in the fruit fly's brain were engineered. Light caused it to take flight in the petri dish.
When I hear about such techniques, I think that the person behind who had the idea to put such a technique in place must be a very creative scientist that has such a long-term vision that's going to help to change the field and the way we think about science. I would describe him as a very visionary scientist who's got big ideas and always thinks one step further than probably most other scientists would do. So I feel it's a great honor to have the opportunity and the possibility to work in his lab to get inspired by this vision that really pushes us to go further and further and further in order to address very ambitious questions and in order to be able to change science.
I think optogenetics is really revolutionary because so before neuroscience was just more of a field of passive observation of neuroactivity of the brain, but now with optogenetics we can actually actively engage or actively interrogate the nervous system and add our own tailor-made activity into the nervous system and see the consequence to other neurons or to the organism's behavior in general. And I think that's why it's so revolutionary, and that's why it transformed neuroscience.
But Miesenböck's ambition doesn't stop here. He continues his research with fruit flies.
I think the technique of optogenetics has matured considerable over the more than 20 years that it has existed now. There are, of course, still areas for improvement, but I have to say I'm not working on these issues anymore. I invented optogenetics because I knew I needed a technology like this to solve some of the neurobiological questions that interested me. One of our main interests at the moment is the neuronal regulation and the biological function of sleep. And optogenetics was extremely important in pointing us to the areas in the brain that are important for sleep and for wakefulness. We've recently uncovered a surprising link between energy metabolism and sleep that puts a physical basis to the long-standing but previously very difficult to test idea that energy metabolism, oxidative stress, lifespan, aging, neurodegeneration and sleep are all somehow connected. I think we may have found sort of the linchpin that connects all these different things.
Over the years, scientists around the world have continued to improve on the technique. After the break, we'll look at the collaboration between German scientists Peter Hegemann and Georg Nagel. The pair together discovered simpler light-sensing proteins from a type of green algae, faster and genetically simpler than the fruit fly genes used by Miesenböck. As Austrian scientist Gero Miesenböck first succeeded in activating nerve cells with light, a German team made a groundbreaking discovery that propelled the field of optogenetics even further. Peter Hegemann of the Humboldt University of Berlin and Georg Nagel of the University of Würzburg. Channel Rhodopsin.
There are two words it is made of, opto, which has of course something to do with vision or with light, actually more with light, and genetics, because there's a gene transfer. So it's always protein you take from one organism, mostly actually from microbes, bacteria, or little algae, and then bring it into an animal, for example. It became very simple since we discovered the general rhodopsin.
Now about 1,000 or 1,500 labs in the world are using general rhodopsin from chlamydomonas for their experiments. And I think the biggest advantage is the ease to use it. It's just one small protein which unifies a channel and a sensor. And the next thing is it's robust.
The team extracted this wonder protein from a simple green algae, Chlamydomonas reinhardti, dubbed by scientists as the green yeast for its powerful role in dissecting biological processes. It all started in 1985. Fascinated by how the unicellular organism moved towards and away from light sources, Hegemann began to study its light perception. Back then, he did not think his attempt to understand this natural phenomenon would lead to applications in optogenetics.
We would like to understand how algae behave, and this is what we use photoreceptors for behaving and orientation in our light environment. And animals do it, and algae do it. They can move and orient to places that are optimal for photosynthetic survival and photosynthetic growth. And this is per se an interesting question. So application is the second part of the research. If we don't search for novel natural phenomena, we have nothing to apply.
This mystery took him over 15 years to solve, and the real breakthrough came when he joined forces with Georg Nagel.
It took a few years. In 2002 we published the first study on generodopsin 1, but this protein was not expressed very well. In 2003, with generodopsin 2, everything changed. It was clearly useful. And then several groups used it, and in 2005 then several papers came out from different groups, all referring to our study, and also they have gotten the DNA, the genetic material they got from us, and could show that it works beautifully. And then the growth was somehow exponential.
After we have proposed that the photoreceptor in chlamydomonas is a light-gated or a light-activated ion channel, the people didn't believe us. So to demonstrate this, you have to take this single protein and express it in some host cells to demonstrate that this is indeed a light-activated channel. And that important step was achieved with Georg.
The science world has a long history of trying to stimulate nerve cells. Back in the 1700s, Italian physician Luigi Galvani used an electrode to pass an electrical current into frog nerves. The action caused the frog leg to twitch, from which Galvani concluded that neurons could use electrical signals to pass information. Modern brain surgery pioneer Wilder Penfield started stimulating the human brain with electrodes in the 30s. Electricity was applied to certain areas of a patient's brain to understand the parts of the body it controls, which allowed him to map the human brain. With the development of optogenetics, cell control can be much more precise, non-invasively, with light. Not only do neuroscientists apply the technique to understand how the billions of neurons function, there are also studies aiming to look for treatment of disease.
There are experiments, therapeutic essays underway, even with blind patients, where they want to bring in generodopsin into the eye. And I think there is a good possibility that this even might help and restore vision in blind people. At least it was shown for mice. And it is probably much better instead of inserting some electrodes or some chips into the eye with lots of electrodes or so. In principle, it should be much more harmless.
You can also apply it for Parkinson's diseases or epilepsy and many other diseases, but it's not so far developed. And the work on application is complicated because you have to introduce the encoding DNA into the cell of interest. And if you want to do it in humans, as everyone knows, this is not free of ethic considerations that we have to overcome or we have to justify before we go into a human cell.
The two teams are now testing new proteins so they can continue to sharpen these techniques.
We want to bring in different tools. This is not only generodopsin, these are also other light-sensitive proteins which either come directly from microorganisms, bacterium or an alga, or which were modified by us and are, of course, light sensitive. I think it's always ideal if they are only sensitive to blue light, so you can still watch your organism in red light because you are not activating the protein. And then with the blue light, you can activate this protein and see what is then the outcome of your activation, of your manipulation.
What we are working on at the moment is to shift the spectrum further to the infrared, because infrared light would be more ideal than visual light, because the penetration in the brain is much higher. Then we combine different tools, and we like to bring into the business light-activated enzymes, light-activated cyclases. This is a gradual improvement. If you want to jump onto a new level, you probably have to change the media. Try to find proteins that are sensitive to ultrasound, or proteins that are sensitive to radio waves. If you achieve this, then you have reached a different level, completely different level. But we need young scientists, maybe coming from different fields.
Inspirations they bring to the field of optogenetics go beyond their discoveries.
He's a very, very nice person. He's an excellent scientist, and I think everybody who is in touch with him feels this. And he has a very, very, on the one hand, relaxed attitude, but also a very deep understanding of science. So when you discuss whatever problem with him, it is always very, very productive and illuminating.
Working with Peter is a pleasure, I would say, because he is an ambitious scientist. He's curious. He has a lot of fantasy, and he never stops halfway, I would say. If he's tackling a problem, he wants to solve this problem, and he wants to bring the solution to an end. He also listening to other people's opinions in a critical way, always open discussions, and that's what makes these cooperations so fruitful.
What I've learned from interacting with Peter, or through understanding how he has decided to run his scientific career, for me this is kind of like a role model situation. Sometimes it's worth it to not just trying to maximize your impact and do everything as fast as possible, but also just figure out what is really interesting for you, and maybe also just take the time to really get to the bottom of it, because sometimes you don't know it, but it will really be worth it. Like in say again, in the case of the use for neuroscience.
Looking back at solving these problems looks easy now, but there were many obstacles along the way. These pioneering optogeneticists summarized their success with a single word: persistence.
After discouraging time period, when you work on something for a long time, you might get frustrated, and you might take your own perspective or life into question, but if you're a real scientist, it comes back quickly. You think about different alternatives and new approaches, and then the enthusiasm might come back quickly. But it goes up and down. It's in every job, probably.
I think I felt more tired when I was young, because it takes so long, and sometimes I thought, I would rather be a bus driver than you know what you are doing, and I didn't see the outcome. And I must say I was very lucky in my later years. I guess the most important thing is that you enjoy what you are doing, and that you find projects and experiments which you like to do, where you want to see what is really happening in nature. And then even if you have to work for longer times or so, then it's not so bad, and it gives you something back.